So good morning, everyone. I'm Yousra Ajouaou. She said I'm a postdoc in the Kirsten Sadler Lab here at NYU Abu Dhabi. So first, I would like to thank the organizers for inviting me to share my data with you today. So in the Sadler Lab, we are interested to study the role of epigenetic regulators in cancer, development, and aging. So let's delve into why we are so interested in aging. Because the global population is rapidly aging, and the World Health Organization predicts that the individuals aged 80 and older is expected to triple by 2050. Beyond the superficial changes that we see with aging, like wrinkles or gray hair, the real challenge is the fact that aging is the strongest risk factor for many diseases, such as cancer, heart disease, Alzheimer's disease, and type 2 diabetes. We face a huge gap between our lifespan and health span. On average, each individual will spend their final 16 years sick, imposing a huge burden on the healthcare system worldwide. So the question now in our mind is, why do we experience health issues as we age? So to unravel this enigma, we have to understand what is the cause of aging, what is the root causes of aging. So the study of aging began in the early 20th century, and recent researchers have unveiled the hallmark that characterizing aging. And among this, they identified senescence, chronic inflammation, genome instability, and most importantly, the epigenetic alterations is one of the primary hallmark of aging. So as we've heard from previous speakers, thank you for this nice introduction to my presentation. Now I don't have to go through all this, but what is important to know is that there is many different epigenetic modifications, such as active and repressive histone mark, some histone variants such as H2AZ, and it came up that the DNA methylation at CpG site is a critical epigenetic marker essential for gene regulation and silencing harmful TEs. So this you heard from the previous speakers. They make up half of our genome, and they can jump from one genome site to another, leading to alteration of cell function, high inflammation. So keeping them repressed by DNA methylation along with other histone marks is essential. So the correlation between DNA methylation and aging has been first done by the discovery of an epigenetic clock. This clock has been developed by Dr. Steve Howard, and it is a mathematical algorithm using 350 CpGs, the DNA pattern on those, to predict any individual biological age that's highly correlated with the chronological age. And the DNA methylation can be influenced by a combination of genetic, environmental, and some stochastic factors. And as you've heard before, the UHRF1 and DNMT1 pathway is primarily responsible for the initiation, but also for the maintenance of DNA methylation through cell division, allowing the transmission of the cell, the DNA pattern throughout the cell. So the question now is the modification of the epigenome a cause or a result of the aging process? To understand this, we need to study how the steady-state epigenetic pattern in young model is organized. And this has been done extensively in the epigenetic field, and in our lab, we are using the liver mouse as a model. So this study has been done by Chi and Filippo here in the audience, and they characterize the various chromatin states in the young mouse liver. And they show that there is 6 main states, but the most striking finding of this study is that 85% of the genome is only marked by CpG methylation that represses over 88% of the total DNA. So this finding, total TE, sorry. So this finding emphasizes the significance of DNA methylation in regulating the liver's genome. The liver. is a primary metabolic organ, and it has the unique capacity to regenerate. So understanding how the epigenetic landscape changes with aging become obvious to us. And we know that as we age, the liver undergoes several changes, such as increased expression of TEs, increased inflammation, and also an augmented senescence. Senescence is defined as a irreversible cell cycle arrest that states usually when the cells cannot divide anymore, they just died. But these senescent cells are resistant to death. So then they start to release deleterious factors for other cells. Then, on the other hand, we see a decline in the metabolic function and also a decline in the regenerative capacity of the liver. So the question now is, what mechanism contribute to the decline of the liver function with aging? And in our lab, we are mostly interested in the regenerative capacity because we know that this capacity decline with age, and this is really a human characteristic. Interestingly, the mouse here, just below the human, exhibit a regeneration pattern similar to the human. This shows that mice is an ideal model to study liver regeneration. So how do we study liver regeneration in mice? We use a well-established model called the 2/3 partial hepatectomy, or PH, where we cut 70% of the liver, and within several days, it regrows to its original size. This amazing process has been studied extensively, and in our lab, in a previous study in our lab, we show that in the early stages of liver regeneration, we see an increase of a lot of epigenetic regulators, and particularly gene-related to the DNA methylation machinery as UHRF1 and DNMT1. So as mentioned before, UHRF1 is critical to maintain DNA methylation during cell division, and its loss in the whole mouse body is embryonic lethal. So we know that UHRF1 is essential for life in the mouse context. Then we wonder if UHRF1 is also necessary for the liver regeneration. That's where our study started. So my main question is, what is the role of UHRF1 in liver regeneration during aging? So to do so, we use a mouse model of hepatocyte-specific deletion of UHRF1 called UHRF1 hep knockout. Those mice are completely normal, except in the liver, we have a completely loss of UHRF1. Using this mouse model, I addressed three main questions. What is the role of UHRF1 in the young mouse liver regeneration? The second part, we will be interested in how the loss of UHRF1 impact the liver phenotype. And finally, I will address how UHRF1 impacts the old mouse liver regeneration. So the first part, we wanted to answer the question, does the loss of UHRF1 in the liver affect liver regeneration in young mouse? This beautiful work has been done and published in Cell Development a few years ago. It has been done by Sami and Chi, two alumni of the lab. They show that the young UHRF1 hep knockout is completely normal in terms of their appearance, their body weight, and the liver looks fine. But the most interesting findings of this paper is that the UHRF1 hep knockout mice exhibit an enhanced liver regeneration after partial hepatectomy. And the expression here, as you can see, of all pro-regenerative genes are expressed in early stage in UHRF1 hep knockout mice. So this findings challenges our previous assumption about UHRF1 in the liver regeneration, because we thought first that UHRF1 may be essential to induce liver regeneration, but according to this, it is not. So to understand the molecular mechanism at play, they perform DNA methylation and transcriptomic analysis. And not surprisingly, they see a huge hypomethylation at the global genome, but also at TE regions. And the most surprising is that despite a global methylation, they see no transcriptional changes in terms of genes, but also in terms of TE expression. So this So this observation raises question about compensatory mechanism at play. Indeed, they show that there is an increase of the repressive histone mark H3K27-ME3 in the TE's regions. So it might be the reason why TE are not expressed in those mice. So to summarize this first part, I would like you to know that although UHRF1 is highly expressed in the early stages of liver regeneration, it is not necessary for this process. Moreover, they show that there is an epigenetic compensation by H3K27-ME3 in the UHRF1 HEP knockout mice that protects those mice from TE activation, but also they lead to an increased pro-regenerative genes at early stages, leading to an enhanced liver regeneration. I would like to close by thanking all my lab co-workers, especially Kirsten for her support and giving me the freedom to study my questions. Also a special thanks to Filippo, Shashi, James, and Shade for their help on the highly demanding partial hepatectomy experiment. I would like also to thank Mark, Nizar, Mohammed for their help in the RNA-seq analysis. Also, thank you, Dipesh and Mohsin for allowing me to use the open field. A big thanks goes to the CGSB organizers, of course, and especially Inas, Susan, and Hanan. Thank you for organizing this symposium. I'm really happy to speak this year. And also, I will encourage you to go to see all my colleagues' work. This is their poster number, so please go. They will show you their wonderful work on epigenetic regulators in different models. And also, if you want to talk further with me, please come tomorrow. I have a poster number 74. And finally, I would like to thank you all for your attention. Thank you.